Hi, I'm Erin Cooper.
And I'm Bonnie Lexizawat. We're from the Department of Biology at the University of Kentucky. The purpose of today's experiment is to conduct a urinalysis and to understand what positive values may indicate. We will have 3 experimental groups: a water group, a control group, and an isotonic group.
Urine samples will be collected every 30 minutes throughout the lab period. You will measure fluid volume, conductivity, specific gravity, and chloride content in the urine samples. Thus, you will be able to calculate the urine flow rate as well as the chloride content in the urine samples.
Okay, to measure the conductivity of the solution, urine, use the conductivity pod, which is connected to the lab probe, and here's the conductivity rod. On the desktop, open Lab Chart 7, go to Setup, Channel Settings, change the channel to 1, press OK. To the right, go to Channel, Conductivity Pod, and change the settings to the most sensitive settings. Press OK. To begin recording, press Start. As a gauge, you can use distilled water, and the conductivity should be 0. Then place the conductivity rod into the urine, and you should see the reading on the screen. Okay, the refractometer is used to measure the specific gravity of a solution. In order to measure the specific gravity of the urine, take a little drop with the droplet and place it on the surface right here. Close the glass covering and look into the end of the refractometer. To focus, move the blue knob at the end, and where the blue line is the specific gravity. You can check the different specific gravities of each different experimental group.
Okay, so once you've collected your sample of urine, you'll take one of the chem strips, which actually measures the different components of urine. So just take one chem strip out. And then there's different little pads on here that will tell you a positive or negative reading. So what you're going to do is just dip it completely in urine. Drag the chem strip against the beaker so you take off the excess urine. And then you're going to look at the different components in it. So what you're going to do is compare it against the different components that are in the urine. So you look at glucose, bilirubin, ketones, specific gravity, blood, pH, protein, urobilinogen, nitrates, and leukocytes. So what you do is just place the urine and then look for the same corresponding color, and then continue through all the different components. And this is also used clinically. So next we will be measuring the chloride content in the urine. You'll be given a little dropper here that are 2 milliliters. So each little dent right there is half a mil. So what we're going to do is collect half a mil of the urine sample and then put it into the test tube. Next, with the new dropper, you're going to take 1 drop of potassium chromate and place it into the test tube. So it should stay a yellow color. And then what we're going to do is drop silver nitrate, drop by drop, until the urine turns into a brick red color. So what we're going to do, make sure you count the number of drops that you place into the test tube. And then continuously swirl the test tube around. So that's 2 drops. 3, 4. And it should turn a dark brick red. Okay, so that was 13 drops. And then what you're going to do is times the number of drops, so 13 in this case, by 0.61 to get the chloride content in milligrams per mil. And that's how you do the chloride content of the urine. So the urine that was tested has to be thrown away properly. So what you're going to do is pour the urine into the hazardous container because it contains the chromate.
So we just showed you how to conduct a urinalysis and to understand its results. Also, to note the differences within each group of specific gravity, urine flow rate, and the excretion of chloride. With the data we collected, one can better understand if ADH was stimulated.
When conducting this experiment, be aware of the amount of urine placed on the refractometer to measure the specific gravity. Also, remember to clean the equipment after each use. So that's it. Thanks for watching and good luck with your experiments.